Welcome. This is the video on what you're going to be doing for your lab assignment. This is lab assignment number 5. I'm going to scroll down to our tab. Here's our lab, and then if you've watched the video on the lab portion for this week, it'll be up here at the top. So I'm going to go down to our assignment. Here we go. So now we are, and this is where the video would be. Okay, now we're in. So as always, I would make this a little bigger. So this is a great presentation. Tissues and skin, they kind of go together. Skin is the integument system. So this will be the first of the organ systems that you cover in anatomy and physiology. And from here on out, it's just all organ systems. So as always, I want you to click through. I'd like you to read through this. There is actually a little bit on homeostasis on how tissues can help regulate body temp, things like that. Great animation here on a little bit about the background on what tissues are. Tissues are groups of cells that have a common function. And part of learning anatomy is the study of tissues. And you will do this throughout A&P2 as well. So there are 4 groups. Here is epithelial, and each tissue is grouped, well, there's 4 major groups. There's epithelial, connective, now they have nervous here, and then muscle. And I'm going to go back to epithelial. Within this group, you have what are called tissue types. Simple squamous epithelium, simple cuboidal epithelium. So read through these sections. It talks about the groups. There's some knowledge check here. I think you should do those, like what red cells are what type of tissue. They are connective. I think that's good to do. And then here's a little bit on the integument system, which is skin. And same thing, I'm going to read through all of this, the layers of skin. There's an epidermis, dermis. There's actually 5 individual layers to the epidermis. Another knowledge check. Which skin layer is not always present? It's this one, stratum lucidum. It's only found in the palms of the hands, soles of the feet. And you're going to go through some of the key parts. Now, once you get down to the bottom, you're going to do a little exercise with receptors on your lab assignments. Here's a quiz. I'd like you to take it. I think it's great practice. And then we're going to go down to assignments and procedures. So first thing, here is a slide viewer. Now, what you would do in lab is you would look at these through a microscope. Now, you're actually ahead of the curve here because in real life, microscopes are being used less and less. Now it's more computers and the big 4K monitors. There are still microscopes being used, but not as much as in years past. Now, here are the 4 groups. So I'm going to click on epithelial. Now, this little equals thing will tell you a little bit about each tissue. So once you go through, I'm going to click on next. And then this is a lung section, a little cut of a lung. These are alveolar sacs within the lungs. They are lined with a tissue called simple squamous epithelium. So once you click on the equals, there we go. It will tell you what tissue type is located here and a little bit of information about that. I think it's great. And then you would go to the next one. See, here are some, it says nuclei. You're going to click on this. This is also some simple squamous. Click on the next one. This is simple cuboidal, little cube-shaped cells on here. And anyway, you're going to have all different tissue types throughout this. And then you will go through each major group, like here is connective, same thing. Go on that. Here are some adipocytes. This is adipose. Click on the little equals, and it tells you a little bit about each tissue. So that's what you're going to be doing for histology. Now, examination of skin, same thing. It's going to pull up. Here's a skin slide viewer, both areas. Click on skin, go to next, click on the little equals. It'll tell you a little bit about each section of skin. This is actually a really good one here. Here's the epidermis. Here are the layers. This is called the stratum corneum. That looks something like what would be the stratum lucidum. And then granulosum, spinosum, and the very bottom is called stratum basale or basale. So go through these, look at them. And then here's some sweat gland distribution. And in fact, this is a point, at this point, I'm going to go back because I do have a lab sheet for you. And what you're going to do is go through and actually look at each section and then fill in your lab sheet. Let's go back out. Okay, sorry, let me go back in here again. Go down, there's number 6. Okay, so here we are. So what I just showed you, it's kind of like you've done what you've done so far. Go through this, look at all of the material that's there. Now, here is your lab answer sheet. And click on that. Now, I'm going to make this a little bigger. Same rubric as always, pictures with lab, do the lab assignments as instructed. It's mentioned this before, but do not open your fetal pig. On the part of the assignment, it says on number 5, we're not doing that. It says something about open up your pig and look at the skin. Please do not open your pig bag up yet. Keep it sealed up. We're going to do that for muscles later. Here are the questions you're going to answer, as always. Pre-labs, they're worth 2 points each. What's the function of nervous tissue? All that information is within the assignment sort of lecture / reading part you're going to do. Now, it's asking you here, microscopic examination, what kind of tissue is this? You would actually write this in here. Oops. If you want to make it neat, you can. Either way, it doesn't matter to me. So you're going to ask what type of tissue. You're going to go through and answer these questions. Like that, to me, looks like adipose. There's some reticular. Here's some, looks like elastic cartilage. Here's some muscle tissue. Tissue looks like cardiac. So go through and type in what you think these tissues are. There's some tissue and skin, additional tissues. So that's pretty simple. And then answer these post-lab questions, like what's the difference between simple, stratified, and pseudostratified? Simple is one layer. Stratified involves many layers of cells. Pseudostratified actually means false layers. There's only one layer of tissue. And it's called pseudostratified because it looks like there's many layers. Things like that. Go through, do good detail. I'll give you full credit. Go through your microscopic examination of skin. Answer the labeled parts of skin. You have a nice picture of that. What layer is it? Papillary layer, reticular layer, things like that. Answer all these questions. Now, this part, sweat gland distribution. You are going to have to take a picture for this, and I'd like you to do the right anterior forearm. So what you're going to do is, let me go back out. You're going to take some betadine. This is actually a really cool thing. And I go to the bottom. Here we go. It says carefully measure and cut out 4 5 centimeters by 5 centimeter squares of graph paper. Should have some of that in your lab kit. Locate regions on your right anterior forearm, right palm, right anterior thigh, right anterior foot. Do not have to have, that do not have large creases. What you're doing is you're going to measure your sweat distribution here. Use betadine swab to paint a square on each selected region. The square should be slightly larger than the graph paper. Allow betadine to dry for a minute. Use the tape to secure a piece of graph paper on each painted square and let it sit there for 20 minutes. After 20 minutes, remove each graph paper and then you should be able to count dots on each square. It's going to represent how active the sweat glands are. It's actually a pretty simple experiment. Put a picture of your right forearm for the bed if you want to get full credit for the exercise. And to remove betadine, go through and then, you're going to wash it off. And then here you're going to enter your distribution on how many dots you find on each square. All right, that's pretty simple. So there you go. Table's worth 5. pictures were 10 points. And put them under here. Oop, right there. Take a picture with your cell phone, camera, upload them, and then save it to your document and put them right there. Here are some post-lab questions, and then here's the last thing you're going to do. You're going to make some little skin calipers. I'm going to go back here. They're going to look something like this. You're going to make some with different lengths. It says make a 1, a 1.5, 2, 2.5, 3, 3.5, 4.0 centimeters apart. So use the paper clips, within your lab kit for this. You're going to need a partner with this. If you don't have a partner, try to do it on yourself. What you're going to do, we do this in lab. We don't do it anymore. We used to. It's called a 2-point test. The way skin receptors work, the closer together the 2 points, the harder it is to tell it's 2 points. The further away you get, the easier it is to tell. Like, I'm willing to bet 1 centimeter you're going to feel like you're only having one thing touch your skin, which you see it here, it's actually 2. So what you're going to do is test sensitivity in different regions. I believe it says, start with the largest caliper and then go down to the smallest. Let me see, middle 4, okay. There it is, sorry. You're gonna test the right and left sides of the body. Try to do all these areas. It won't take you very long. Just do a quick 2-point test, and you're measuring to see can they feel 2 points or not. It's very simple, and you're gonna write that. It's basically kind of a yes or no in your graph here. It says observe any sensory differences between the left and right sides of the body and record your results. Now, I'm willing to bet that if you're right-handed or left-handed, you may feel a difference in the two of those. So fill this in. This section is worth, I want to say this is 15 points. And I'm going to go back to our lab answer sheet. There it is. You're going to fill these in, and you have 2 post questions with that. And that is everything we're going to do for lab number 5. Now, this is not due until the end of November. So you got plenty of time to do this. And we are going to have a couple more labs between now and then. And that's when they're going to be due. The end of November is right before the semester ends, and I'm going to have to grade everything. So go through, read the instructions, and email me if you have any questions. Thanks.